This is Unit 6A, Part 2.
This is a lymph node. It's an encapsulated lymphoid organ. It has a connective tissue capsule around it. You can see here on the diagram, that's all connective tissue capsule right there. Now the lymph nodes are located along, they're sprinkled throughout the body all along the lymphatic vessels. And they act in a way as filters for the lymphatic fluid or lymph, which is moving along the lymphatic vessels as they collect fluid from the as they collect interstitial fluid from the body, and they can carry in a solubilized form any cell debris or foreign molecules that may have gotten into the person's body. So you have lymphatic vessels leading in towards a lymph node, and here you see a number of incoming lymphatic vessels, which are called afferent lymphatic vessels. So here's a number of them, and they allow the lymph to move into the lymph node itself. Notice that there's two outgoing drain points or two outgoing lymphatic vessels. These are efferent lymphatic vessels carrying lymph away from the lymph node. There are fewer efferent lymphatic vessels than there are afferent lymphatic vessels, which means that there is some slowing down of the flow. Also, the lymph node itself is much larger, and there's much more space for it to actually fill with fluid. So the fluid slows down and percolates all through here and can come into contact with the millions of immune cells that are present. The lymph node itself has these accumulations of lymphoid tissue. These are called follicles. Here, each of these round circular areas are called follicles. These are particularly rich in B cells. Each of these is a follicle. The inner part, like many organs in the body, the inner part is called a medulla. So this would be the medullary area, all in here, all through there. And the outer part is the cortex, and you find in the cortex, that's where you find the follicles. There are other types of lymphoid cells in there, in the tissue. There are macrophages, particularly, because macrophages are particularly good at taking in foreign antigenic molecules by phagocytosis, chopping them up, and then presenting them on their surface. So that's a way of presenting foreign antigen to a lymphocyte. So, as I said, the lymph nodes, these are principal and very important lymphoid organs of the body. These are secondary lymphoid organs. Remember, the bone marrow and the thymus are primary lymphoid organs. They have a capsule. They're all along the lymphatic vessels, and you have some that are near the surface of the body, particularly the inguinal lymph nodes in the groin, the axillary lymph nodes in the armpit, the cervical lymph nodes that are around the head and neck. And these are closer to the surface, so they can be palpated, they can be felt. If a person has a infection, say in their left arm, then the infection, and the infection, let's just for as an example, is due to a bacteria. Well, the bacteria are in their arm multiplying, and some of the molecules, soluble molecules, are coming off from those bacteria and getting into they're in the interstitial space, and they get carried into the lymphatic vessels, and they move along, and they have an opportunity then to interact with all the millions of lymphocytes and macrophages in the lymph node, and maybe, hopefully, and it usually does happen that way, that they come into contact with some lymphocytes that have the appropriate receptor on their surface. So with activation, with presence of an infection, typically you get increase in size of the lymph node because cells are dividing, cells are being activated. You get some inflammatory response by macrophages and even more macrophages there, and so a lymph node can get enlarged, and that's what a doctor does when she's palpating or feeling, the lymph nodes, the regional draining lymph nodes. So for example, you have an infection in your hand, the regional draining lymph nodes some of them would be present in the armpit, axillary lymph nodes, or if you have a sore throat, your lymph nodes in your neck, underneath your jaw or on the sides of your neck, could get enlarged. And finding a large lymph node does indicate that there is an infection going on. So in the lymph node, you have the opportunity for interaction with specific immune cells, receptors on the T cells and the B cells, so the T cell receptors on T cells or antibody molecules on B cells can interact with antigen. So it acts as a kind of like a filter on that lymphatic fluid or lymphoid fluid, lymph, for antigens and allowing for an ideal environment for immune response. There are two basic functions for lymph nodes, and I've mentioned this before. There's filtration. The lymph node filters the lymph, and there are macrophages in that lymph node that can phagocytose and destroy and chop up the foreign molecules or foreign bacteria and microorganisms, and not only kill them, but also, and I said this earlier, the second thing is they can present antigen. They present those foreign molecules on their surface, and it's an ideal way of activating whichever T or B cells inside the lymph node has the right receptor for that particular antigen. So it's a site for an immune response. All the necessary cells are there. Incoming fluid that has been percolating through all the interstitial fluid and getting into the lymphatic vessels is entering through the afferent lymphatic vessels, bringing it into the lymph node, an opportunity, and it slows down in there because it's a bigger space, and also there's fewer draining efferent lymph nodes. So it's a perfect ideal environment to get an immune response. Structure of the lymph node, as I said, well, it's a round or bean shaped, has a fibrous capsule. There are supporting connective tissue structures. They're called trabecula, and they divide it up into compartments. The inner part or inner region is called the medulla, and the outer region is called the cortex. And you see these trabecula, there are these walls here. These are the trabecula here, these wall areas. So giving the structure some support. And you see the follicles of lymphoid tissue, lymphoid follicles. If there is activation of one of these follicles here, for example, it gets much, much bigger, much more dense because the cells start to divide, so it gets bigger and darker. And that's part of the reason why it can be palpated or touched, felt, that there's an increase in size. Okay. Of course, throughout they have these reticular fibers that also provide a framework. Macrophages and lymphocytes are on those fibers and ready to phagocytose, and if there's any microorganism, to kill microorganisms and process the foreign molecules and present them on the surface of the macrophage, so it presents antigen on the surface, presenting it to lymphocytes. The outer cortex is where you have the follicles. If you have an actively proliferating, multiplying, in other words, it was activated by a specific antigen area, the follicle becomes what's called a germinal center with many, many B cells in it. I mentioned that just earlier. You also have some macrophages that are a macrophage lineage cell that's even better at processing antigen, chopping it up, presenting it, and they're called dendritic cells. In the deeper cortex, out in the follicles, there's mostly B cells, and deeper in the cortex are the T cells. And T cells can move between the lymph node and blood and lymphatic fluid. They can exit the lymph node, they can come back to the lymph node. And then there's those trabecular cords, medullary cords extending. No, I don't know. Let's not talk about this. Okay. There you go. That's the structure of a lymph node, and you have many, many lymph nodes in your body. The fluid, as I said, comes in through the lymphatic fluid comes in, or lymph comes in through the afferent lymphatic vessel and flows underneath that capsule, so it flows into the subcapsular sinus, or the space or sinus beneath the capsule, subcapsular sinus, travels into the smaller sinuses or spaces inside the lymph node. The fluid will exit into efferent lymphatic vessels because they're fewer. The lymph stagnates or slows down in the lymph node, allowing time for interaction between the lymph and any antigen in it with macrophages and lymphocytes. Okay, let's talk about other lymphoid organs. We did talk just now quite a bit about the lymph nodes, which is a very important lymphoid organ. There are many of them in the body, but let's talk about other lymphoid organs. We did talk about the thymus, which is a primary lymphoid organ. It's the site of maturation of T cells. There are all the other lymphoid organs, well, the bone marrow and the thymus are primary. All the others are secondary lymphoid organs where cells that are mature and have come out of the primary ones then go to the secondary ones, and that's where you can get an immune response. And they include lymph nodes and the spleen. The tonsils are in your throat. You have quite a few pairs of tonsils, not just two, but I think there's 3 or 4 pairs. I've forgotten. We'll see in a second. You also have pairs patches in the small intestine, particularly in the ileum, the third section of the small intestine. And then throughout your body, wherever you have connective tissue that's typically just near some surface, some area of your body where there's a lot of interaction with the outside world, like your gut, GI tract, or in your lungs, you have lymphoid or lymphatic tissue sprinkled all through that, just underneath the epithelial tissue or even in the epithelial tissue. They all have the reticular fibers as a supporting meshwork. Okay, let's look at the thymus itself. The thymus, as I said before, is just above the heart. It has 2 lobes, so it's a bilobed organ. It is a primary lymphoid organ. It's the site of maturation of T cells. It's where the T cells become mature and immunocompetent. So this is where they mature to immunocompetent cells. It does secrete hormones, but those hormones act on the T cells inside the organ itself. And in many books, it is incorrectly described as an endocrine gland. It is not an endocrine gland. It is a primary lymphoid organ. It does produce hormones, but they don't get secreted into the blood. They act locally only. The organ is not part of the endocrine system. The organ is not a gland. It is a primary lymphoid organ, site of development of T cells. Size, I said this earlier, varies with age. In infants, it's quite large, and as people get older, it gets smaller and smaller. It atrophies. It's called thymic atrophy. It gets smaller, gets reduced in size with age. Here you see the positioning of the lymph node. There are the 2 lungs, there's the heart, these 2 lobes hang down. This is a slice through a thymus. Did I say lymph node? This is the thymus. Thymus, there are the two lungs, there's the heart, there's the thymus, a bilobed organ. This is just one lobule, one section of it. The inner lighter part here that you see, that's the medulla. And the outer part with many, many, many more cells, so therefore more nuclei and therefore darker, is the cortex. The new T cells are multiplying heavily inside the cortex, but they mature in the end inside the medullary areas, which are further in, deeper down. That's all medulla. You find these unusual cells called Hassel's corpuscles in the medulla. So an outer cortex, an inner medulla, many, many cells in the cortex, fewer cells, and Hassel's corpuscles in the inner medulla. It's not, the thymus is not like the other lymphoid organs. It is a primary lymphoid organ, strictly for T lymphocyte maturation. It does not respond to foreign molecules, to antigens. Foreign molecules cannot get into it. It is a site where T cells mature. It does have reticular fibers. It does secrete hormones, but they act locally, and they help the lymphocytes, the T cells, become immunocompetent T cells. It's for this reason, because it was earliest in the, well, its function, I think, was described in the 1970s, but at first they did find that there were hormones, and for that reason it was incorrectly described as an endocrine gland. It's not. It's a primary lymphoid organ. Okay. Let's talk about the other ones here, the tonsils. I think there's 3 pairs of tonsils in total. There's the thymus. Spleen is on the left side of the body, just dorsal and lateral to the stomach. So this is the spleen. Peyer's patches are found in the association with Peyer's patches is also, it's a secondary lymphoid tissue. They're collections of, unencapsulated collections of follicle filled with lymphocytes. They're found in association particularly with the ileum. You also have an appendix hanging off of the, right at the beginning of the large intestine, and that is also has a lot of lymphoid tissue in it, in the structure. I'm going to leave the cartoon for you to read. You know, people don't know often what the spleen does. In fact, and I'm sure I have it on a later slide here, what the spleen really does is act as a filter on the blood, because blood enters and percolates slowly through it and can interact with lymphocytes there. So if there's any foreign molecules, foreign antigens in your plasma, it will have an opportunity to interact and activate immune cells in the spleen. Okay, so the spleen is the largest lymphoid organ. Again, a secondary lymphoid organ on the left and back behind the posterior or dorsal to the stomach. That's not that important. It's a site of filtration of blood. It surveys the blood. Immune surveillance is when the immune system is checking. So the tissues, the interstitial space, is monitored because the interstitial fluid is draining into lymphatic vessels and then moves along until it comes to a lymph node. So that the surveillance is done at the lymph node. In terms of the blood or the blood plasma, the surveillance is done in the spleen. I mean, you do have immune cells within your blood that can also get activated if they come into contact with a foreign molecule, but a lot of the interaction, the opportunity is best in the spleen, where the flow of blood is very sluggish, very slow. It goes in and slows down there. Okay, and with activation of a T or B cell, the cell that has the right receptor, antigen receptor on it for the particular antigen you have in you, foreign antigen you have in you, that those few cells that have the right receptor on them, they get activated. And with activation, with lymphocyte activation, T or B cell activation, you get proliferation. In other words, the cells start to multiply. So you go from just a small handful of T or B cells that have the right receptor to many, many cells. It takes some time. It can take 10 to 14 days before you have enough to have an adequate immune response, but because cells are going to be dividing, say, 12 to 20 every 12 to 24 hours. So once you get through enough doublings, you'll have quite a few cells and a decent number of cells that can respond to that particular antigen. Okay, this is the structure, a description of the structure of the spleen. What you have in the spleen is a blood supply. There are splenic arteries and there are splenic veins. One major splenic artery feeding blood in and a major draining or splenic vein. They divide up into smaller blood vessels. And what you have is that around you have a cylinder of white blood cells, a white blood cell or lymphocyte-rich area all around the arteries that go through. You have many central arteries that are in the spleen, okay? And it's surrounded by a column of white blood cells. And this is truly a terrible, it's a terrible photomicrograph of the spleen. There's much, much better ones. I don't know why they put this in. If you Google normal spleen histology, you should see a diagram that, or a photomicrograph that looks kind of like these bluish circles, and in the center of each one you would have a central vein in cross section. Whoops, doesn't show on top. There you go. So you'd have no, it doesn't show, but in each one you have a central vein, and that's kind of shown here. Central artery, not central vein. Central artery, that's kind of shown. There's a central artery, and this area here, that's the white pulp. That's the area of lots and lots of white blood cells. The rest would be normally, on a good histological slide, would be all red, and that's called the red pulp. So you have these white blood cell rich areas all around surrounding central arteries. Okay, interrupted by a phone call. So you have these circular areas. They're actually a cylinder all around the central arteries. There's many of them. And the rest is red pulp. The white pulp is filled with lymphocytes. The red pulp is filled mostly with red cells and macrophages. Then it's macrophage rich. So essentially what you have is an area, it's quite an open vasculature. I believe it's a fenestrated vasculature, or no, it's a sinusoidal capillary bed. So capillaries are very open and blood can actually get right out, percolate all around, interact with white cells, macrophages, etc. There are other functions associated with the spleen. It also is the site of breakdown of older red blood cells. Red cells don't live forever. They have about a lifespan of about 100, 120 days, and then they get older and they're taken up by macrophages in the spleen and get phagocytosed and chopped up. Any stored iron, and any of the iron associated with the heme group in the hemoglobin will be stored and used, exported out and used by the bone marrow to make new red cells with new hemoglobin in it. The protein part of the hemoglobin, the globin chains, they get chopped up into amino acids. But the heme group gets processed and forms in the liver and gets formed into bilirubin. In a fetus, the spleen is a site of red blood cell production. So in a fetus, before birth, in the fetus, it's not just the bone marrow that's producing red cells, but also the spleen. It is also in young people or adults, the site of storage of platelets. So you do have an extra supply of platelets that can be released if necessary, and they're stored in the spleen. It has a capsule around it. It contains many lymphocytes, particularly in the white pulp. There are macrophages and red cells in the red pulp. And that's said all through here, I guess. A lot of macrophages in the red pulp. Okay. So what other kinds of secondary lymphoid organs or tissue is there? What kind of immune tissue do you have? You have spread throughout your body, you have mucosal associated lymphatic tissue. It is typically not encapsulated, a lot of it, but these are accumulations of lymphoid tissue, lymphoid cells associated with mucosal epithelium. They are mucosa associated, so they're close to mucosal epithelium, mainly in the digestive tract and in the respiratory tract, where there's more chance of coming into contact and interacting with foreign molecules, particularly infectious microorganisms. The tonsils are part of that, malt, and yeah, it's four pairs of tonsils. A lot of people think it's just two. It isn't. It's four. They're all in the back of the throat, even the posterior wall of the nasopharynx. It's a very simple lymphoid organ. Tonsils are partially encapsulated. They don't have a full capsule around them, and that can be seen very easily by the fact that they have this. That is a connective tissue capsule all around this section, histological section of a tonsil, but they have also this crypt. A crypt is a space embedded into the wall of a structure, and you can see here, this is all connective tissue all along here, all the way along here, but then it's bare. There is no connective tissue. There is no capsule of connective tissue. This area here, this crypt, is an opening right into the tissue itself. So antigenic structures that are that can get into here can interact directly with lymphoid cells that are found in again, lymphoid follicles. You see there's a right there's a lymphoid. Let me get something a little darker. Lymphoid follicle there and there and there, and all this is, remember, it looks like it's flat in 2 dimensions, but it's not. It's 3 dimensions. So these spaces here are other crypts that burrow into the inside of the tissue, just like this one here. Okay, so these are all also crypts. Okay. As I said, tonsils have follicles with, if they're activated, if a follicle is activated, you have a germinal center. That's just a follicle that has, it's very dark because there's many dividing cells in it. Again, not fully encapsulated. Crypts can trap and destroy bacteria. So you can get it, you have a chance of interaction with bacteria there. Other aggregates of lymphoid tissue or lymphoid follicles Another example are the Pear's patches. I told you found in the gastrointestinal tract, particularly in the ileum. Similar structure to the appendix, except there's absolutely no capsule at all, but similar in structure with follicles, and they act to destroy bacteria that are locally dividing. You know, the tonsils in the throat are going to be exposed to and get bacteria in them. If you have a bacterial infection where bacteria is dividing in the throat on the surface, some of it will get into the tonsils. The same thing here, where some bacteria, if you have an infection, an accumulation of one particular type of bacteria that can pierce through and enter into the tissue just below the mucosa of the ileum and the small intestine, then it'll interact with these Pear's patches, the lymphoid tissue found within the Pear's patch, and generate an immune response. I'm going to talk to you about memory later. Here's a cross section through a part of the ileum. It shows Pear's patches. These are these lymphoid follicles here. This is mucosal associated lymphoid tissue. You know you're in the intestine because these are, all of this are villi. Remember, again, it's not 2 dimensions, it's 3 dimensions. So the finger-like projections of the villi, in this case, they got cut through just the tops, just the round projection, so it looks like little islands, but really they're all projecting up. They're just not projecting flat upwards. They're, you know, up and curved and in and out of the plane of the microscopic slide. So you get these islands. These are all villi, and here's the Pear's patch just below the mucosa. So it's a mucosal associated lymphoid tissue. This is smooth muscle in the wall of the ileum.
And that's the end of Unit 6A, Part 2.